developed some potency assays. So, for instance, I don't know if that shows up here. Yeah, colors got lost. But basically, you can throw latex beads, for instance, into these cultures, and these RPE cells will eat them, and you can actually show them on EM inside the cell. We also have developed a flow cytometry method where we can actually quantitate the phagocytosis of these bioproducts. So this is great. We can make RPE in the laboratory. They look normal, but what can they do? So we tested several different animal models. One is the Royal College of Surgeons, or the RCS rat model, which is an excellent model for macular degeneration. And when you looked at the visual acuity of these animals that were treated with these human RPE cells, we saw 100% improvement in visual acuity over the untreated controls. Similarly, in the Stargardt's mouse model, we were actually able to show that they could slow the progression of or onset of that disease. But what I think is most striking is the histology. So if you look at one of these RCS rats after 100 days, there's only one layer of these of the outer nucleated cells, the photoreceptors. So that animal was essentially blind. By contrast, the animals that were treated with the human RPE cells, here at the same time point, they had a very robust 5 to 7 layer thick outer nucleated layer. So very extensive photoreceptor rescue. We've also had to develop a manufacturing method that would be acceptable to the FDA for clinical translation. So we have developed actually an assay that allows us to detect a single undifferentiated embryonic stem cell in over 1 million RPE cells. This is very important because obviously teratoma formation is a concern of most of the regulatory agencies. But also, too, it's important to note that our highest clinical dose is only 200,000 cells, so we could detect a single cell that was undifferentiated. And similarly, we've done spiking studies where we've added in up to 10% undifferentiated embryonic stem cells towards the end of the process, and we didn't end up with any undifferentiated cells at the end. So this pathway basically just precludes the remaining undifferentiated cells from being there. We've also carried out extensive safety studies to assess tumorigenicity. We didn't see any safety signals, no tumors, no ectopic tissue, and observed in over 200 animals. Long-term data from spanning the lifetime of these animals revealed no evidence of teratoma formation. And based on this exciting preclinical data, we then proceeded into the clinic. The first trial was dry AMD for 12 patients, and then a second trial, Stargardt's disease, another 12 patients. And the first patients for both of those clinical trials were carried out here at UCLA on July 12th by Dr. Stephen Schwartz, and he will be giving you an update on that. We've also begun to do some studies at other sites in the U.S., and we actually transplanted the first patient in Europe, a Stargardt's patient with these RPE cells. Actually, that was just last month. So, as I mentioned, Steve will be giving you an update on the clinical results. As I mentioned earlier, we're also studying other cell types that could impact visual function. We've been studying what are known as retinal neuroprogenitors. And what we discovered is that they actually can reverse the progression of photoreceptor degeneration. And when you look at the ERG, the equivalent of an EKG in the eye, we actually, for both the cones and the rods, which are the A and the B waves, we saw a very significant improvement in the animals that were transplanted with these retinal neuroprogenitors in both the A and B wave. Equally as important, when we look at the thickness of the outer nucleated layer, it was a very dramatic rescue. So you could see a very healthy, robust outer nucleated layer in the animals that received these RNPs, whereas we saw very extensive degeneration in the untreated animals. And it looks like this may be a paracrine effect, so we're in the process now of trying to isolate what those factors actually may be. Another program in the eye is for corneal repair. There are over 10 million people with corneal blindness. The cornea is the most transplanted organ. Solutions at present are PKP, which is the transplantation of the full thickness of the cornea, and more popular now is just the transplantation representation of the corneal endothelium with Descemet's membrane, which is red. And so we've now been able to create entire sheets of these corneal endothelial cells. And when you look at the various markers of normal corneal endothelium, such as ZO1, which is for tight junctions, you see a very similar profile in the corneal endothelium that we derive from embryonic stem cells. Likewise, when you look at the sodium potassium ATPase pump, you see a very similar profile in normal corneal endothelium versus the ES-derived cells. And I would also like to briefly touch too on another type of cell, the hemangioblast, which is a bipotential cell that has the capacity not only to create the various blood cell types, but also vasculature and endothelium. They look very unremarkable, like a bunch of grapes, but what they do in animals is very significant. So here's a study of ischemia reperfusion injury to the eye, and when we labeled these cells with green fluorescence protein, within 24 to 48 hours they homed right into the damaged retinal vasculature, and you can see them incorporating. By contrast, in control animals and in uninjured eyes, they do not incorporate. So these are very smart cells. They know where to home. And we did similar studies in spontaneously diabetic rats, and again, within a few days, you can see the incorporation of these hemangioblasts into the damaged vasculature. And as you may know, diabetes, all the horrible secondary complications are mainly microvascular in nature, so you're probably getting similar repair elsewhere. And along those lines, we looked at the use of these hemangioblasts to restore blood flow. So this is a Doppler flow for the hind limbs of some mice, and you can see there was no blood flow initially in this animal. After a single injection of these hemangioblasts, we completely restored the blood flow within a month. Similarly, when we injected these into animals that had severe myocardial infarcts, we were able to cut the death rate in half. As I mentioned, in addition to forming the vasculature or capillary structures, these cells have the potential, with just a different culture media, to form all the various blood cell types, so that we've also turned these hemangioblasts into entire tubes of red blood cells that transport oxygen just like normal transfusible blood. We've got these cells to enucleate. This is particularly important because obviously for early clinical therapies, if you're transplanting cells that do not have nuclei, the risk of tumor formation is eliminated. We've been able to turn these into virtually all the different blood cell types. In fact, we have several iPS cell lines now from O-negative patients so that we can create universal blood that matches everyone in this auditorium. We've turned these hemangioblasts into megakaryocytes, which, as you know, are responsible for making the platelets, and we demonstrated that those platelets could actually form retractable clots, and they also incorporated into the thrombic site from laser-induced arterial injury. So, in the next several years, we're hoping we'll be able to bring both the red blood cells and platelets into the clinic from both ES cells and IPS cells. We're also hoping to bring in some therapies that are based on hemangioblasts for vascular repair, again, using both embryonic stem cells and iPS cells. And one more interesting cell type we're working on is the mesenchymal stem cells. There's accumulating evidence that suggests that they can actually migrate to the site of injury, exert immunosuppressive effects, and facilitate the repair of the damaged tissues. And there's some very exciting preclinical data that has been generated. And at present, there are over 200 clinical trials using autologous or allogeneic MSCs that are underway. So why use embryonic stem cell-derived MSCs instead of the adult MSCs? Well, one is you could have off-the-shelf therapy for immediate use. We can create an unlimited source of these cells. Again, these cells are immortal, so you can start out with as many cells as you want. They're very easy to derive, less labor-intensive to generate, and they can be expanded into very large numbers. So this is Wally, a Red Sox fan. He represents the amount of MSCs that you can get from 1 unit of adult bone marrows. From the ES cells, you could fill the entire Fenway Park. So really dramatic, over 30,000 times the expansion capacity as the adult cells. So in addition to being more youthful and living longer, they could have potentially greater efficacy. Indeed, as a control, you often allow these cells to be expanded, and when they're older, they actually can serve as negative controls. And we've started now to test what these cells might be able to do. We studied a model for MS, multiple sclerosis, and this is the onset of the disease in an untreated animal, whereas in animals that received the MSCs, it was significantly delayed. And then just finally, I just wanted to make the point that once we have this unlimited source of tissue, by using tissue engineering techniques and seeding them onto various scaffolds, we have the capacity to create more complex tissues and structures, indeed entire organs. This is bladders that Tony Atala had grown up that were transplanted into patients. And we did some work with Tony actually testing some of the SCNT-derived renal cells. So what you see here is our attempt at a miniature kidney, and we seeded some of these cloned renal cells onto these polycarbonate membranes. This little plastic bag represents our primitive attempt at a bladder. And after you transplanted these back into the nuclear donor animal, in this case it was a cow, we started to see the appearance of the straw yellow material. It looks like urine, and indeed, when you look histologically, you could actually see that the cells had assembled into glomeruli, into tubules, and they actually connected up in a unidirectional fashion with the polycarbonate membrane, and we actually were able to detect elevated amounts of creatinine and urea. So these were removing toxic materials from the system of these cows. And then just very finally, stem cells has a lot of controversy associated with it, and that's often because it involves the destruction of human embryos. And we published a couple of papers in Nature and Cell Stem Cells showing that we could actually do this without hurting the embryo. So in IVF clinics around the world, there's a method known as PGD, where they look for genetic abnormalities, where they can just pluck one cell from the embryo. And what we did is we used that technique. By just removing one cell, we can actually create embryonic stem cell lines that have all the normal markers and do all the exact same tricks. And the biopsied embryo continues to divide and gives you a nice healthy embryo that can be frozen down. So we now can create embryonic stem cells as well without destroying the embryo. And then finally, we'd just like to thank the people who really did all the work, and in particular, Steve Schwartz. Thank you. Thank you.
Sense of what a privilege it is to work with Bob Lanza, just extraordinary scientist. I'm just going to switch, hopefully, to my talk. Get a little bit of luck here, please. Invite your talk file.
Any purple label that you changed.
Sorry. I tried to put a macro in. So as we get started here, let me, you have to go right back through these. I apologize. The perils of two talks. Thank you for your patience. I have to say that working with Bob has been inspirational, and that didn't work, did it? One more time. I thought I had it selected to slide forward.